Good afternoon, distinguished professors and friends. Welcome to the inaugural webinar of ACRT Frontiers Forum, Decoding Aging, Decoding Cancer. My name is Rachel, and I am the Managing Editor of Aging and Cancer Research Treatment, ACRT. On behalf of our Editors-in-Chief, Editorial Board, and the Editorial Office, I would like to extend our warmest welcome and sincere appreciation to our distinguished speaker, moderator, panelists, and everyone joining us online today. For our first webinar, we are delighted to welcome Professor Yuxuan Lu from the Southern University of Science and Technology as our invited speaker. Professor Lu will present today's lecture entitled Accelerating Gerotherapeutic Discovery, High-Throughput Screening, AI, and the Future of Healthy Longevity. We are also delighted to have Professor Ting Ni from Fudan University serving as today's moderator. Following the lecture, there will be a discussion section, which we have invited four outstanding experts to share their perspective on emerging advances and future directions in aging biology, gerotherapeutics, and healthy longevity research. We hope today's webinar will stimulate insightful discussions, inspire new ideas, and foster future collaborations across our scientific community. Before beginning today's scientific program, please allow me to introduce ACRT briefly. Aging and Cancer Research and Treatment. Please wait a moment. Aging and Cancer Research and Treatment is the official journal affiliated with Shenzhen University Medical School and is published by Science Exploration Press. The journal follows a rigorous and international peer review process and is published quarterly. ACRT is co-led by Professor Xin Zhixu and Professor Baohua Liu as the editors-in-chief. Under their leadership, ACRT is committed to advancing interdisciplinary research at the interface of aging and cancer, while providing a high-quality platform for basic, translational, and clinical research. Since its launch, ACRT has been fortunate to receive strong support from many renowned scientists, such as our associate editors, Professor Jun Huang from Zhejiang University, Professor Zhiyong Mao from Tongji University, Professor Zhengpeng Lou from Mayo Clinic, and Professor Vincent Gurley, as well as we have distinguished advisory editors, including Professor Vera Gobunova, Professor Steve Hovers, Professor Steven Kowalczkowski, Professor Guanghui Liu, Professor Zhao Qi Wang, and Professor Gerd Pfeffer, Professor Yu Fangshi, and Professor Zhongjun Zhou. Their support has been instrumental in shaping the journal's scientific vision and international development. Today, ACRT has an international editorial board comprising 52 distinguished scientists from 14 countries. Many of them are internationally recognized leaders in their respective fields, including Professor Chunlong Chen, Professor Jin Ke Cheng, Professor Lun Zhidai, Professor Yi Bingkang, and Professor Zheng Yujun, as well as Professor Guido Kramer, Professor Chuanyuan Li, and so on. Although ACRT is a young journal, it has been honored to publish contributions from internationally recognized researchers, including Professor Baohua Liu, one of our editors-in-chief, Professor Jiankang Zhu, together with today's speaker, Professor Yuxuan Lu from the Southern University of Science and Technology, Professor Song Tao Shi from Sun Yat-sen University, Professor Jin Donghan from Peking University, and Professor Si Wang from Capital Medical University. From an outstanding editorial leadership to a distinguished editorial board, and now to contributions from many leading scientists in aging community, ACRT is steadily established itself as an international platform for high-quality research. We believe that an outstanding journal should be more than a venue for publishing scientific discoveries. It should also serve as a bridge that connects researchers, inspires collaborations, and accelerates scientific progress. Now it's my great honor to introduce today's moderator, Professor Ting Ni from the School of Life Sciences, Fudan University. Professor Ni is a leading researcher in cellular senescence and aging biology. His work has advanced our understanding of the molecular mechanism of cellular senescence and has contributed to the development of innovative technologies and strategies for senescence identification and intervention. Professor Ni, thank you once again for joining us today and for your support of ACRT. It's our great honor to have you with us. The floor is yours. Okay, I will stop my
Thank you, Rachel. Thanks. Good afternoon, everyone, and welcome to the inaugural ACRT Frontier Forum. And I am Ting Ni from Fudan University, and this is my pleasure to be your moderator for today's webinar. We are delighted to have you with us as we kick off the ACRT Frontier Forum webinar series. Thank you all for taking the time to join us. And I hope today's presentation and the discussion will spark new ideas, encourage engaging conversations, and provide an enjoyable opportunity for academic exchange. And it's now my great pleasure to introduce today's speaker, Dr. Yuxuan Lü from Southern University of Science and Technology. Dr. Lü's research focuses on aging biology and gerotherapeutics, with major contributions to high-throughput screening, AI-assisted drug discovery, and the development of interventions to promote healthy longevity. Today, he will present a lecture entitled Accelerating Gerotherapeutic Discovery, High-Throughput Screening, AI and the Future of Healthy Longevity. We're also delighted to be joined by 4 distinguished panelists who will take part in the discussion following the presentation. They are Dr. Yonghan He from Kunming Institute of Zoology, Chinese Academy of Science, and Dr. Yang Li from Fudan University, Dr. Xiaotao Shen from Nanyang Technology University, and Dr. Yudong Zhu from Hangzhou Normal University. And a warm welcome to all of our panelists. We look forward to an engaging discussion later in the section. With that, Dr. Lü, the floor is yours.
Thanks a lot, Professor Ni. It's my great honor to have this fantastic opportunity to be here. So again, thank you to the ACRT editorial team to organize this event. So the next 30 minutes, I will briefly talk about what our group has been doing since a couple of years. And I'm very happy to have an opportunity to give you an insight, not only the biology of aging, but also the importance to develop potential protection to extend health spans. So here's we briefly use a different to explain the cutting edge multimodal platform for drug discovery, from aging mechanisms to translations. So first of all, I would very like to show you a graph, which I always show to all the important people. And as we might know that aging is a global challenge, because we can actually see that the last couple of decades, our percent of elderly, so the people older than 65 years old, have been significant increase compared to 50 or maybe several years ago. More striking, if we look into the line, we actually that this trend is more pressing in China. So China, from now on, I say that from 2020 or 2023, the acceleration of aging process actually is very dramatic increase. And we know that now our government, but also society, has already taking a lot of measure to to support the research on aging, but also the economic. We need to quickly adapt to the aging society. So that's the reason why I think the aging is very important, and we need to find a way to solve it. First of all, I'd like to show you two examples. So the left-hand side, I guess many of us already know the hallmark of aging. So this is the newest version of the hallmark of aging, consists with 14 different hallmarks. But more importantly, this figure actually is the age-related disorder and disease to the aging hallmark, but also aging itself. So as a gerontologist, I always believe that the majority of age-related disease, namely like including like a cancer, cardiovascular disease, but as well as metabolic disorder, sarcopenia, and some of the disorder including like skin aging, hair loss, and as well as the like ocular disease. So those things are more or less caused by biology of aging. So that means when we get old, we will have a higher chance to have those diseases. That means if we can have a way to slow down or even prevent our aging process, we might hopefully able to stop those diseases, right? So on that side, the paper we published together with our former colleagues in Max Planck Institute for Biology of Aging in Germany. So at that moment, we used one of the very typical or classical anti-aging candidate we call rapamycin. So rapamycin, as some of people might know, that is immunosuppressor drugs initially used to organ transplantation, but now at least in the animal models, we saw that when we feed rapamycin, for instance, like mice or the other animal, it can significantly extend the health span, but also the lifespan. And what we found in 2022, which is highlighted by the Nature journal, we found that even we used the short period of rapamycin administration, we could still get a significant lifespan extension, at least in flies and mice. So that's actually give us a very important point that for the development anti-aging, or now they call the gerotherapeutic aging, I think that is something we could do, and we could success hopefully in the foreseeable futures. So that's the first statement I would like to make to everyone, that is the intervention to extend health span, hopefully also lifespan, is as important as to understand the aging mechanism. I think this is two sides from one point. We need to understand the aging mechanism. In the same time, we also need to discover the intervention to extend the health. So regarding the discovery of gerotherapeutics, I think the greatest example happening is the ITP program. We call the intervention testing program, supporting by the National Institute of Aging, so in the United States. So this project has been run over 20 years, using mice to test what drug can slow aging and also extend lifespan in mice. So since the last 20 years, it has been tested more than 16 different drugs, and including the rapamycin, and also some of the supplements, including like fish oil and MMM. Obviously, some work, some doesn't. But the point I would like to make here is not the program itself. Obviously, the program is excellent. But on the other hand, I don't think anyone wants to wait another 20 years to test a 60 drug candidate. And at the end of the day, maybe you only get one or two drug that has potential to extend the lifespan in mice. But that doesn't mean it's happening in human, right? So we need to find a way to accelerate this process, because as I showed you in the first slide, we are getting into the aging society, and including me, I also want to find a drug that can extend my health span as soon as possible. So that's the point. It's a couple of ago. So the field started to think about if there's any feasible that can accelerate this agent drug discovery process. So this comes from the Matt Cabling, so one of the railroad aging scientists in the United States. So groups mentioned that there's a way to use a short-lived model to accelerate this process. So here's what we like to show you. And in our lab, the test, so Southern University and Science and Technology, we started to integrate to apply those ideas into our lab. So we're trying to build a pipeline from the beginning to the end. So we focus on the preclinical study. So because now we know that AI but also the short-lived animal actually can accelerate a lot of the discovery process, drug discovery. But also, if we also want to talk about the development, we might need to use animal model to accelerate this process. So here's just to give you an example. Another example we just recently published on the Aging Research Review, I think it was in April this year, and we together with people working on silicon, fish, but also working on the AI and also the other field which related to aging, we wrote code to talk about the high-throughput screen aging and related disease drug discovery. So we identified there's a way that we can use the AI automation together with the short-lived animal, as I already mentioned, the silicon, the tooth fly, Drosophila melanogaster, as well as the renewed aging models, fish, so the Africa turquoise fish. So those animals, since they live only weeks, couple months, half a year, so those animals are suitable to the early age drug screening specific for the husband, also measure the lifespan case. And also these things can together with the recent very powerful AI integration. So the AI can help us to analyze the high content image, analyze the behavior data, but also the physiological matrix. So at the end of the day, the AI together with those animal results can give us a very powerful result to tell us which compound would have the high potential to extend the health span, also give us all of things that we hopefully can use for future mechanistic side, also biomarker discovery. So here I would like to give you a few examples. So the first example actually is happening in, I think, 30 years ago, so 1/3. So it's our collaborator called Professor Shen, when he was, I think it was in Beijing University. So he developed the first generation high-throughput screening for C. elegans. So he called the warm farm. So it's very basic high-throughput nematode culture system. So he generated a chip that you can put the C. elegans or worms into a very, very tiny chamber, and then you can use the microscopy to track the behavior and also measure the lifespan and also the health it looks like. And obviously it's very fancy in that, I mean, just 10 years ago, but now obviously now they have a more advanced technology. So here's the new versions from Professor Shen, which I would like to show. This work has been published in 2024. So as you can see now we can actually integrate into AI tracking. So when the worm move, or you can actually use the camera, the movements, so you know how they eat, how they interact with the other, and also how much, I mean, how much they walk. So in that case, you can actually assess not only the survival, so not only death or life, but also their behavior at the young stage and also the old age. So in that case, actually, it's possible to use they call the worm CNN to assist and also establish the the aging process, to which gerotherapeut will be able to extend health span. But of course, I think the worm is one case, because when you look into the world, you will see that a lot of very cool model organisms, including worms, but also the fruit flies and also the fish and all the others, right? So I want to show you the fly stuff, because there's a room for that. Because fly, I mean, I always say the fly, it has brain. I mean, it has brain, it has legs, but also flies. But more importantly, they also have a social behavior, including fighting, trying to play with their couples, and also we have sex in that. So, and where you can also see that, unfortunately, is in Chinese, but I would like to explain you quickly. So, it's a couple of behavior change with a similar to human. So, for instance, the movement in the fly will decline with age, as similar to human, but social interaction also change with age. That is also quite similar. And we also found that nowadays the sleep for flies, the fly get old, the sleep fragmentation also increase. So means those things are very similar to humans. And for flies, because you can also culture in a very small place, and you don't need any animal welfare regulation, which is very good for the high-throughput screening for the drug candidate. So here is what we do. We build up a fly tower. It's an AI powered longevity discovery platform. Now is located in the Sussex in Shenzhen. And we try the automate platform. So use the robotic arms, use the standard cell culture plate, which for where flies, and also use this camera to record the fly behavior. And data we are able to test more than 100 different drug candidates in a couple of weeks, so we can give the health score, but also the lifespan score. That's that. And at the moment, what our platform can do is we can assess the motor, can assess the sleep, vehicle, so represent the gut health, the fertility, but also the lifespan. And in the future, which I'll talk about later on, we are all looking to the health. So I think that's very important, particularly in the society at the moment. So here I'd like to give you a few examples of what our screening platform are doing. So one of the things that developed by our collaborator and students called Huaizhen, and he created this very fancy flight plate platform on 96 well plate. So which he can do 96 different drug candidate screening just in a very different manner. So I'm not going through to but I guess you already get that that's very tiny and also very efficient to do the testing. And we also use the same platform to assess the feedings. And we know that because, at least in the aging field, we always know one very powerful lifespan and also health span extending intervention is dietary restrictions. So to assess the thing, we are able to see how the fly eats and whether those compounds which extend the lifespan would have effect on the food intake. So we can identify and also isolate the confounding effect that might caused by the changes. And we also can use the similar platform to integrate with microscopy, so we can try to also measure the fly fertility. So just counting how many eggs they lay, and then just trying to use the YOLO algorithm to test them. So this is actually straightforward, and all of this result has been published on the self-reported method last year. And moving on that, we want, since we already have, so we want to test whether really working for good. So first of all, we try to test. Well, we try to take some positive drug candidate. So we took list from the drug A. So the big database for aging-related drugs, which contain more than 100 different compounds, and we use this to test to run, and we found that despite that most of the candidate extend lifespan, which already shown in the drug age form, but all of them can have a strong benefit health span. So we test different parameter, including like locomotor, figure A and figure B is locomotor. We found that only few of the drug can have a benefit to make the age flies walk longer distance compared to the control flies. But we also found that at least around 15% of those candidates would have benefit on the max speed. So that means that even despite they might not walk very long distance, they will have observed their muscle activity, so they can have a very sudden max speed increasement if you have stress. And we also check some of the drugs. It has a very good change to sleep better, including the rapamycin as a control, and also the green tea extraction, so e.g. CG, and of course the others. So here I show you a brief example. I would like to impress you that this platform is very helpful because the important thing is like, as we might know that the lifespan and health span is not always correlated, as we know that actually now getting live longer and longer, but health span is not catching up. So using this system, we are able to not only screen the drug for lifespan, which is like the IDP they have been doing, or their main primary endpoint is the lifespan. Our platform are able to run the health span in parallel. So here I give you an example, and trying to measure the compound, and we're trying to find out if the drug benefit health span and lifespan in the same time. So as you can see, this is a Q1 square, but we also found that there are also some drugs that can extend the lifespan but not health spans. And more interestingly, what we found is also there are strong sex differences in terms of the drugs. So some compounds benefit female more than males and the other way around. So I think this is very important information because this actually gives us very strong as information that can use for the next drug development. So that's something I would like to give you briefly. So, and since I'm trying to switch a little bit scale, a little bit direction, because I not only drug but also the diet is the key drive of healthy aging, right? So nowadays there are a lot of study, of course, on the vegetarian diet or DASH diet or something like a plant-based diet and so on. So we know that because there's a lot of very useful content in this diet, so there's also a way to test them. But unfortunately, because for mice, because diet is very complicated, it's very hard to do the kind of high-throughput screening to test which compound or even maybe which ingredient in those diets has a strong benefit to the plant. So that's the reason we're also trying to see if our platform are able to at least partially solve this issue. Yeah, so we took very nice very nice stuff we just recently with the Professor Zhu Jiankang. So this is very cool stuff project I would like to give you a brief. So we used editing approach to biofortify tomato. So namely that we are able to increase the of important supplements, or you can say the compounds in the tomato, and then make super So I hope that you are able to see that the DHC, the vitamin D3, and also the GABA, lignin, and carotene, and also lutein, so, and vitamin C. So all of them has increased a couple of fold significantly. And we believe that this super tomato will have a greater health benefit compared to wild type tomato. And therefore, we use our platform to test this to run this experiment. And we test so we test oxidative stress, we test the sleep quality, notion, but also lifespan. And I hope you can see that so compared to control, some will see that the wild tomato might have a little bit benefit compared to control. So in all this testing, but the BT, so the biofortified tomato, has a very significant improvement in oxidative stress, but also sleep quality, locomotion, as well as the lifespan. So that means if our platform actually are able to test this complicated diet and also vegetable and whatever. So I think this is a very powerful tool that we can also multiple things that link to the health span extensions. And now I will also doing something which is still unpublished or still under development, but I would like to just share with all of you briefly. So I hope that create something more important. So the first thing actually came to my mind, it was, I think, 5 years ago. So many of us are still in quarantine at that stage because of the COVID-19. And when I was isolated, I read the paper published on Nature. So it says like the chronic social isolation signal starvation very strong impact on sleep in social lives. And then in 2020, there's also a couple of papers, including those ones that say that the social attraction in life is regulated by the muscularity and the rotary systems. So given that the fly is very good model to do the social isolation, attraction, and happiness study, so we now also trying to build up a platform, actually a module to screen the fly behavior. So this is the first version I want to show you. So we are able to create very tiny Y maze, which we know that usually is very typical behavior testing for mice or the other animal. But the fly, we can also do that, and we are able to identify that this is the memory actually decrease with age. So as you can see, the young and old, obviously the young move more than the old, but the working memory. So what I'm saying is the alternation rate, so whether they turn left. So if young, they remember left or right, so they will decide to do some similar in the next round. But if they don't, they what they did before, they will 50-50 randomly do whatever they want. So that is very simple, but I think it's also a very powerful approach to prove fly can use for memory testing, and in the future also trying to do more complicated behavior testing, like a social attraction and also social isolation with us. Okay, so I think more or less today I still have a few slides which I think is very important because as I said, for drug factor discovery, and we cannot only do things in the lab. I think it's very important to also try to find if there are ways to move things towards to translation or application. So now we actually started to run the spin-off, and we establish automated platforms for efficient translation. So as you can in software, we already started to automated our XML. So include the robotic arm, including the drug pipetting system, but also including the automated filming systems. So all those things actually are able to increase our throughput, but also more importantly, and also increase our efficiency and also standardize everything, which is hopefully that pitch to the pharmaceutical industries. So and now we already have, so this is the final, hopefully the second final slide, which nowadays I do a lot of pitch to our potential investor, and we are doing beyond to the academic, the basic research. We're also trying to find out the way to collaborate with company, the Cimeris, which we have been started our collaborations since 2023, and this is based in but when I moved back to China, we also started to collaborate only the pharmaceutical company, because aging is not only drug thing, right? There's a lot of things, as I already mentioned, like supplements, and also maybe the cosmetic. They also want to find a way to slow down skin or organ or whatever aging. So, and when we to China, when I back to China, we also go with a cereal, Maru, also a couple of companies that's on our list. So we're trying to collect the data, and hopefully that in the future we are able to create a aging phenotypic platform that help to accelerate the drug discovery. So last but not least, I think I would like to share with you this what I think is very important. So we need to, if we want to breaking the oldest challenge for human being, that means solve the biology of aging, we need a new mindset, and obviously we need a new technology, so including like AI or this high-throughput stuff. And obviously at the end of the day, we also need the new model, and also trying to integrate all those things to make our life better and happier. So yeah, I just stop here, and thanks everyone for listening, and I would like to take all the questions from the audience. Thank you.
Thank you very much, Professor Liu, for such an excellent and inspiring presentation. We now come to the discussion, and we will spend about the next 15 minutes exchanging ideas. So we have a list, and maybe Johan can raise the first question.
Okay, yeah. Can you hear me?
Yeah.
Oh, yeah. It's a wonderful talk, and he just introduced his work from the Western discovery to and to the high-throughput the screening system, and it's fantastic. Yeah, just wondering, actually, model is very important, and obviously this is the first time to learn about the drug screening use the food flow. In our lab, we usually use the cell system, and this is the classic system, of course. Yes, I'm thinking that maybe model is very important, of course. Maybe I'm not sure whether Dr. Liu. So according to drug screening, any candidate drug can be transferred to, for example, the mice. Do you detect anyone? This is the first question, because I think model is very important. Yeah, fruit fly is very important, but mice maybe the most used mice model. And how about the second? How do you evaluate the side effect, especially when you evaluate the effect of the compound at the fruit or chemicals, and if extract it from the natural right?
Yeah.
So this, I think these two questions may be very interesting.
So thanks a lot, Professor He. I think it's very important questions, and I got these questions. I think not every day, almost every week, people ask me the same question, because the new model is very important. We need to embrace the new. We also need to acknowledge that we don't know much about that, right? Compared to the mice or the other standard discovery. I mean, the pharmaceutical company used the very standard pipeline. So So there's two things I would like to briefly. So first off, I think we have to look into the biology, right? Because all the animals are based on the biology. And as you know that aging is not the single
Yes.
disease. So aging is very
Yeah.
conserved. If
Yeah.
you can see that from Sierra Leone or even from the very small, they are aging more or less, at least phenotypically, more or less the same as mice as a human, right? The movement
Yes.
they will decline. The family, so the ovary or the testes actually decline, and if they have a brain, for instance, like also have a decline, or the vision decline. I can tell you that there was a study vision, also the hearing or decline with age similar to human. So and in that case, that's we say that since age is very conserved, so at least if we're going to find the drug and systematically delay aging, we believe that it could have the translational rate should be not very low. So that's the first point I would like to make. And then second point is like as I told you that I tested I did replication in a very long time, and so I also test a lot of different compounds, including forming, spermine, and also a couple of very famous anti-aging drug candidates when I was in Germany. And at the lab, I was there, was a leader, so she had a big lab, and we are able to not only the experiment in flies, actually we do most of the experiment in mice in parallel. So we collect a lot of know-how, and we know that at least for a drug which is target the metabolic, so the immuno system that conserve life. So that's what we have. I mean, since what we collect the last 10 years in the lab. So even that, we of course we believe that those things are going to happen. But of course, I'm not to say that we need to have the data. So what I can actually all the compound, not sorry, all the those compound, the top three and all five compound we found from the slide, we are actually doing experiment in mice, and we can see that at least more than 15% of show very strong benefit in mice. So which I mean, I'm not expecting 100% of this, but we have more than 50%. I think this is already very good sign that if yes, then we can actually reduce the usage of mice, at least for the early stage. Because I mean, I still believe that the mice is very important, and we have a lot on mice. But the thing I would like to break, we should not use mice to test the candidate as that, because it takes a lot of time and you get very few candidate, which you potentially we can move to human, right? So if fly can offer us a quick approach to get enough candidate that we can up to mice or even potentially testing in the big animal, including like a dog, pig, with no human primate before we apply to human.
Thank you. Thank you very much. Another question, just for discussion, because as you also mentioned that age is not a disease, okay? Even in China or in the USA, WHO do not define the aging as a disease. So usually when we try to test the effect of a compound, actually we usually focus on age-related disease, right? For example, the diabetes, the cancer. Even in the United States, the first a clinical trial for anti-aging used medicine. They started from 1915, I think, almost to 10 years. The result come out. Actually, so it's a engine for the field that we usually talk. We want to therapeutics. So usually very challenging because, as you said, it's not things. I think
Okay. Yeah, so I think I want to maybe just quickly reply to you about this. And I have been asking, I mean, there's a lot of people ask this question, and I think
Yeah.
people working on this, including you, also got a lot of this question, right?
Yeah.
And there's a couple of approaches now that can try to, I say, circumvent this. One of the very good approach is taken by the in silico, I guess you might know that, because that was the almost a unicorn on the AI drug discovery. And I think Alex Taiwan Wong, he is basically longevity biohackers. So I believe that at least what he told me that most of the disease it comes from aging, but his
Yeah.
company actually think disease, but they have a very ancient approach that's called the dual purpose, which is very important. So we have a drug that can cure age-related disease, but the basic mechanism why they cure the age-related disease is because slow down the biology of the
Aging.
process.
Yeah,
So
right.
I guess this is the way we can try to work with that, at least this moment.
Yeah.
But nowadays we also know it's like a GLP-1.
JPL agonist,
So
right?
they claim that they will be first longevity drugs. I guess it's the time or just compared to consider that the overweight.
Yeah.
20 years ago, no one believed that overweight is disease. I mean, of course, it's not a healthy situation, but it's not disease. But now you can
Yeah.
develop
Right.
a target overweight. So I think that's the point I would like to make.
Yeah. Dr. Lee.
Thank you. Maybe okay. Can read next question. Thank you.
Thanks Lee for the talk, and I also actually very interested by several aspects that you discussed. First of all, I would want to share or maybe discuss about the participant you emphasizing, because in my lab I was also doing elegance, and you are expert in your flow field, and when we are doing experiment, we also focus on not only lifespan but also health. But when I was doing, I'm curious that what do you think is best parameter features to define what is the healthy span? Because our movement, I was doing egg laying like you did, and sometimes not always consistent. So I'm allowed to hear about the aspects about the definition of health span.
Wow, that's actually a question, right? And so I would say it depends on what kind of parameter you can collect efficiently. And at least for worms and also ideally for flies, I think the locomotions is probably the best you can do. See how they move, right? The camera now they actually can track, and also but it's not everything, right? And but if you learn from mice and also even from human work, and I think there's always a term called frailty index. You know the frailty that I'm sure you know frailty index. The frailty index comes from multiple parameters, and then they put together and make a frailty index. I think aging is a systematic, and I guess it will be very important to also do maybe frailty index like approach for citizens, and we can maybe have a radar map for with the movement and egg laying on the other. So if the candidate intervention can improve all the parameters, so the frailty index or this is very good, right? But we might also see that some of the candidates improve one parameter or maximum two parameters. So we still say that it improves some specific health span parameter, but not everything. So I think that we could do it together. Maybe in the future, we also need to have something in a systematic way.
Yes.
See, we already want to do it, want to push this short-lived animal together to use on the stage. I think we need standards as something. Yeah.
And another thing that I noticed, you are using your high throughput platforms for drug screening, and we all know maybe many people are interested in genes that longevity genes or anti-longevity genes that maybe also could be a good thing to test in your platform. Have you did and
so that's a very good question. So I haven't done that unfortunately because we don't have enough resource at the moment to do stuff together. But I mean, this platform is able to do this, and we are thinking to produce maybe some cancer or Alzheimer disease and disease because it's very popular. I think it's also in flood, but also in silicon trials. And I think many silicon and also scientists are already used to do the drug testing, and so he would like to go better or couple people. So I think it is feasible. It's right. And the platform I just showed you is kind of concepts because I believe that maybe the people can see the concept, they can do what they prefer to do or they believe is important. I also think that the longevity gene obviously is very important, and to understand the mechanism and for like hopefully the health span using the gene therapy or the other approach. Yeah.
Fantastic. Thanks for the talk. That's the question.
Good.
Thank you, and thank you, Yuxue. Next, we can invite the Xiaotao for raising the question. Xiaotao, are you here?
Yeah, I'm here. Yeah,
Thank you.
Hey.
Yeah, hi, Yuxue. Nice to see you again. Yes,
Yeah.
so good talk. So I have questions about the drug screening. So you mentioned that you use the high-throughput platform for drug screening for the longevity or for the health span, right? So a question that just like you're mentioning is not one simple disease like other diseases like obesity or cancer. I think the aging should be very complex. And so now for a lot of people they think sometimes for the disease is not one drug or one target, right? Sometimes things multiple target, multiple drugs, right? So I'm not sure, do you have ideas if you use your form those multiple drug combine drug together for the disease for the aging drug screening?
Yeah, very good question. So the short answer is we already started to do this, and the first choice we take is the traditional Chinese medicine because it's very complicated, and we collaborate with our industry partner who provide who select the combinations based on the historical Chinese medicine book and because the Junchen Zhuoshi. So we try to find it and try to test, and we do found we define the conditions it works sometimes far more better than the single compound, and yeah, I think that's make a lot of sense. And like also, if it's when I was in Germany, we were trying to compare mice with the other drugs, and we also see it's kind of like a addictive effect on the lifespan and also the health extension. So I think that's important because back to what you mentioned, that the field has been testing different aging intervention and I think more than a couple of decades. But the most powerful intervention so far to the dietary restrictions, right? And the reason for that, because I guess dietary restriction is not one single intervention, it's a kind of combination, different diets, different compounds, and I guess that's a way we really want to push our extension into this approach. And I think I've put, as long as we have high enough food, we can do whatever which we believe is feasible. Yeah.
Okay, I see. It's great. Other questions.
Thank you.
Thank you, Xiaotao, and thank you, Yuxue. We have Xudong. It's Xudong, are you here? If you have any questions, yeah. Okay, thank you.
Sure. So Thanks a lot. Firstly, you gave us a very good talk, and I would summarize it as this technique drives the innovation. And I'm quite impressed by your AI plus flu platform, and also there's a fly plate, and also very impressive with your tomato. And I have relatively aspects to think about this issue because flu is a model, but it's not like the animal, like the mice, Professor Herman, and also different as the human beings. So would you like to comment how it translates into individuals, human beings, how the findings can help us to have these issues?
Yeah, actually, that's a very good point. And I think, let's say, so part of the things I already mentioned as I already replied to Professor He, I think, previously, at the the biology of aging is very conservative. It means so the single pathway and we identify in humans also is implied or is in mice. So it's like the baseline for translation, right? On the other hand, and we also use AI and also use in our lab, we also have mice, but also animal models, which I didn't have the time to tell you, but we are doing things parallel. So we collect the data not only from but also from those animal models. And I can tell you that the fruit fly can give quite strong heat at the moment. Yeah, but I think that this is very important point that hopefully next time that I have time to share those comparisons like I just randomly like 100 candidate to test in mice and then do it in mice and also do it in any model and see how this success rate from to mice hopefully to be. I think this is something we need to have a very solid scientific data to prove my theory, this conservative biology of aging theory is true. But I think very good point. Thanks a lot.
And also there's another issue because we're investing in agents. We want to find the market for the hallmarks, but sometimes they are primary causal regulated, but sometimes they're just correlative biomarkers. So, okay, in the fruit fly, they are more like behavioral aging marker, right? So, can you find out what's the really primary or secondary biomarker they are really involved in and how to intervene them?
Good. So that's a good question. So I didn't have time about that. So actually, we collaborate with the other flights. I mean, before I think found in Guangzhou, but also in somewhere else, and we are able to trying to label the important genes with P and also the other fluorescence proteins. So we're trying to see that by drugs extend span, whether those genes also activated or silenced using the use fluorescence to light up. So we're able to also use to equip fluorescence camera to see how it goes. So obviously, this is not the way we can see that, to get all the information. At least we see that we can have information only the phenotypic information, but also some key genes, some signal pathway, how the activity they change under the drug conditions. And now also consider to come with the high-throughput RNA screening. High-throughput RNA-seq, because not expensive nowadays, so we can just raise to the RNA-seq and even the multi-omic approach to get the molecular in combined with a big cloud. And so I hope that in by this approach, we will have a more comprehensive overview to understand how the drug or candidate are working for the mechanisms. Okay.
Yeah, it's really fantastic. And we'll see next generation of Cineline your lab can produce. Okay, thank you.
Xudong and Yuan. Actually, I have a question from the audience. And the question: how can AI efficiently integrate multi-omics data to improve the identification of novel therapeutic targets? Yeah, and this is for
Wow, this is very large, very broad question. So AI obviously can do a lot of things, but if we look to the drug discovery, the drug discovery pipeline, mainly the very early stage. So I would say the virtual screen, right, to try to find out whether the compound we talk about small molecule, how the structure, whether it fits to maybe one or another proteins or genes. And if you're going to do gene therapy, you can also use AI design more higher efficient genes gene target, the gene target that's able to gene or activate gene more efficiently. So that's nowadays, if we look into most of the AI drug companies they're doing. But what we believe, what we found in our platform, say that the AI also integrate into other steps, say for drug discovery pipeline. In our case, is a preclinical stage. So we integrate the AI. So we the behavior movements and also the other the other phenotypic parameters. So that we ask the AI to help us to identify whether it's moving or not. With the fans or not, because obviously human eye cannot track everything, right? But the AI able to, as long as you can capture them. So this is something very powerful, and I was that know before. Thank you.
Okay, thank you for your wonderful answer. All right, and then to Dr. Liu, Dr. Peng Jun, and to all our panelists for the discussion, and to everyone who joined today. We hope you found today's webinar inspiring, and we look forward to welcome you to the next ACRT Frontier Forum. Thank you, and have a wonderful day. Thanks.